There are three parts to the Calvin cycle: carbon fixation, reduction, and regeneration. In carbon fixation, 3 CO2 molecules enter the cycle and combine with 3 REBPs by an enzyme called Rubisco to create 6,3-BGA. In the next stage, reduction, ATP and NADPH are used to turn 3-PGA into G3Ps. One G3P will then leave the cycle to become glucose, and in the last stage, regeneration, the G3Ps are rearranged into REBPs with the help of ATP. An important thing to remember is that it takes 3 cycles to create one G3P, and it takes 2 G3Ps to create one glucose molecule. Plants have developed structures called stomata that are able to control water loss from the leaves. On hot and dry days, the stomata close so that the plant can conserve water. When the stomata are closed, O2 builds up in the plant, leading to oxygen replacing CO2 in the Calvin cycle in a process called photorespiration. There are 3 types of plants that have evolved to try and avoid photorespiration. One is C3 plants, which fix CO2 into a 3-carbon compound in the Calvin cycle. These have their stomata closed frequently and are prone to photorespiration. Another type is C4 plants, which separate carbon fixation from the Calvin cycle by having it located in a different cell in the plant. They fix CO2 into a 4-carbon compound with the use of an enzyme called carboxylase. The last type is CAM plants, which separate carbon fixation from the Calvin cycle by having it occur at a different time. At night, the stomata open and let CO2 into the plant so that it can fix a 4-carbon compound. In the day, the stomata close and CO2 is released into the Calvin cycle.